The overall goal of this procedure is to register single-channel activity and to measure intracellular calcium concentration changes in cystic monolayers freshly isolated from rodent models of polycystic kidney disease. This is accomplished by first flushing the PCK rat kidneys via abdominal aorta in order to remove blood from the renal tissues. The second step is to isolate the epithelial monolayer of the cysts. Next, the isolated monolayer can be used for conventional patch clamp recordings of ion channels in the apical membranes of epithelial cells. The isolated tissue can also be used for loading with calcium dyes for quantifying intracellular calcium concentration changes in the epithelial cells. These parameters measured in the native environment of cysts can be compared to healthy tubules in order to study the process of cystogenesis.
The main advantage of this technique is that it allows studying ion channel activity and calcium flux in the renal cysts. This analysis provides physiologically relevant data obtained from freshly isolated tissues.
With this approach, we can monitor single-channel activity in the freshly isolated epithelia. It is a unique method because it can be used to precisely characterize ion channels' behavior. Using different modifications of the patch clamp technique, we are able to test channels' response to various drugs in cystic and in healthy tubules.
The freshly isolated cyst monolayer can be used for intracellular calcium imaging. This technique allows pharmacological screening and functional studies of intracellular calcium homeostasis in cystic cells, which are important means to discover new treatments for the prevention of polycystic